So what is operon? Operon means what? O-P-E-R-O-N. Operon. Operon is a set of genes which are present adjacent to each other. A segment of DNA. You can see this is a group of, a set of several genes which are present adjacent to each other constitute operon. Structural genes, adjacent to structural genes, operator gene represented by O, adjacent to structural genes or cistrons. Structural genes may be 1, 2 or more than 2. Adjacent to operator gene, promoter gene, P, promoter. Adjacent to promoter gene, regulatory gene represented by small I, regulatory gene. So regulatory gene, promoter gene, operator gene, structural genes, terminator gene. This is a set of genes which are located adjacent to each other. It is a group of genes, it is a set of genes called operon. This is the operon in the bacterial cells, E. coli. Regulatory gene I is transcribed into messenger RNA, and this messenger RNA is always translated into regulatory protein, either repressor or activator. Regulatory gene I, it is transcribed into messenger RNA, and this messenger RNA is translated into regulatory protein, either repressor or activator. And structural genes are transcribed into a messenger RNA, and this messenger RNA is translated into proteins or enzymes which are required by the cell to perform special functions. This is the operator gene. This is the promoter gene. Promoter is the gene to which the enzyme RNA polymerase binds and transcription starts. RNA polymerase binds to promoter, then it moves along the DNA to allow transcription of the structural genes. Structural genes are transcribed into mRNA, and this is catalyzed by RNA polymerase. Okay, and for that, RNA polymerase must bind to promoter. It must bind to promoter gene. This is the operon which is found in E. coli bacteria, E. coli bacteria. So what is an operon? Operon is a segment of DNA that comprises one or more adjacent structural genes, an operator gene, a promoter gene, and a regulatory gene, small I, regulatory gene. Regulatory gene is represented by small I. Operon works in cooperation with a chemical substance which is called repressor protein or activator and inducer substance. And operon always works in cooperation with repressor or activator, which is a regulatory protein, and inducer substance. In 1961, two French microbiologists, Francis Jacob and Jacques Monod, at the Pasteur Institute, Paris, proposed a mechanism which is called Operon hypothesis. Operon hypothesis in 1961 by Jacob and Monod at the Pasteur Institute for the regulation of gene expression. Operon hypothesis for the regulation of gene expression as a result of their studies on the metabolism in E. coli. And operon acts as a gene regulating unit. Jacob and Monod got Nobel Prize in 1965. Inducible operon. This is the operon which is switched on when a chemical substance called inducer is present. Inducible operon means this is the operon which is switched on when a chemical called inducer is present. The scientists have found lactose operon or lac operon in E. coli. Lac operon or lactose operon is an example of inducible operon. Lac operon or lactose operon, which is found in E. coli, is switched on if the inducer is present. It is switched off when the inducer is absent. What is inducer? Inducer is a chemical, a substrate, or any metabolite which inactivates the repressor by modifying the structure of the repressor. Repressor is the regulatory protein produced by regulatory gene I. Inducer is a chemical substance, a substrate, or a metabolite. Inducer inactivates the repressor protein by modifying the structure of the repressor. In lactose operon or lac operon in the E. coli, lactose is the inducer. Lactose or its isomer, allolactose, serves as inducer in the lac operon of E. coli. Lactose or its isomer, allolactose, serves as inducer in the lactose operon in E. coli. Lactose is the inducer. It inactivates the repressor by modifying the structure of the repressor. Presence and absence of inducer will determine whether the operon will be switched on or switched off. The inducer joins the repressor and forms repressor-inducer complex. This complex with modified repressor now prevents the repressor from binding with the operator gene of the operon. This frees the operator. This keeps the operator free so that the enzyme RNA polymerase can move from the promoter to the operator gene and then to the structural genes. Now the structural genes are transcribed forming a piece of messenger RNA, which is polycistronic because several genes, when transcribed messenger RNA, it is called polycistronic. The messenger RNA is translated into polypeptides or enzymes. The enzymes are called inducible enzymes. The inducer substance, substrate molecule, or a metabolite, it associates with the repressor protein so that the repressor protein cannot bind to the operator gene. Okay, so operator gene remains free. Operator gene lies adjacent to structural genes. When the repressor does not bind with the operator gene remains free because inducer is bound to the repressor, modifies the structure of the repressor and forms inducer-repressor complex. The modified repressor is unable to bind to the operator gene. Operator gene remains free. It is not bound. It is not obstructed by repressor, which is a regulatory protein. This frees the operator gene. Now RNA polymerase. The enzyme RNA polymerase binds to promoter, then it moves to the operator gene which is free, then it moves to the structural genes. The structural genes are transcribed into a piece of messenger RNA. Transcription occurs. This messenger RNA is now translated into enzymes. Translation occurs. And these enzymes are called inducible enzymes. That means structural genes are expressed. The structural genes are expressed. They have formed the enzymes. They have formed the polypeptides. That means the operon is switched on. And because inducer is present, in presence of inducer, operon is switched on. The structural genes are switched on. The genes are expressed. This is the role of the inducer.